Oops. So we need to think about the exam date. Today's Wednesday, and if you look on the syllabus, the first exam is next week. I checked with the classroom scheduling people, and it looks like next Wednesday would be a time when you can come in early. There's no class before us, according to what they said. Has anyone noticed? There's no one in here when we walked in today, right? So I think they're accurate. So what I'm thinking is I could come in a little early. That would give you time to just relax and slow down a little bit so that you don't feel rushed. And it seemed like most of you were raising your hands with that option of being able to come in a little bit early. So what I usually do is get here by like 1:45, and get set up, and then if you can make it in, great. I don't design the exam to be longer than a normal lab period, lecture period. It's just, I want to give you some extra time. I'm thinking next Wednesday. All right, so that will cover topics most likely through today's lecture on transpiration. I have a few last things to say about that. And I'll probably draw the line there, so to speak. But I'll double check that and let you know for sure on Friday. Questions about the exam right now? Okay, I'll make sure to get that on your calendar. We left off here, I think. So with regard to transpiration, the idea of thinking about, if you work up a sweat and then how does that sweat evaporate off you, under what conditions? It's a pretty good analogy. We looked at this idea that with a leaf, they have that still layer of air molecules called the boundary layer, and that it can be thick or thin depending on where they are and what's going on at the time. Certainly wind will thin out the boundary layer, which increases transpiration rate. Plants can adapt pretty well to their environment in terms of how to try to slow down transpiration rates if they're in a habitat that tends to really put a lot of pressure on rapid transpiration. They can adapt. And so one idea is just reducing their leaf size so that their surface area is smaller, and that helps reduce transpiration rates through their body. The shape as well, I'll talk a little bit about that. Leaf hairs, we have the idea of, I'm sure you've noticed some leaves look fuzzier or hairier than others, so those trichomes. And these are two desert adapted plants that are displaying that. They look fuzzy, they feel soft and fuzzy. And so basically what that's doing is it's helping trap humidity, water molecules near their leaf. It's also messing with wind flow so that it disrupts wind flow, keeps your boundary layer thicker. And so it's actually a pretty good strategy to try to slow down their transpiration rates. Because if you're desert adapted, as you can imagine, it's going to be kind of challenging to get day-to-day water easily accessible to your roots. Here's an interesting idea with leaf shape and also where they put their stomata. This is actually a leaf, believe it or not, and it is in cross section. What you're seeing here is the top of the leaf on the top of the image. And if you look along there, this is under the microscope, and you can see, it doesn't seem to be really any stomata on the top. And then if you look along the bottom here at the very, very bottom, there's no stomata there. So it's like, is this leaf holding its breath? What's going on here? You have to look in. So you go in to this tissue that's got these invaginations like this, they actually call those stomatal crypts. I don't know, interesting name. So those stomatal crypts. And so if you look carefully, if you know what stomata look like under the microscope, you start to see some, like, so right here and here, here. Here's one, that's a nice one. I'll circle that one on the image here. Where'd it go? Here it is. So those two kind of darker cells are the guard cells, and then the pore. And so if you start looking in the crypt area, you see that there are those stomata. And so they've actually kind of tucked their stomata away. In essence, what that allows is you get some nice high relative humidity in these trapped air spaces here in the crypts. Plus, you've effectively really widened your boundary layer, if you want to think of it that way. You have a lot more distance to get the air molecules all the way out of the leaf. And so effectively, you're ramping up the thickness of your boundary layer. And so this is a pretty extreme example of adaptation. So again, you have some stomata peppered out throughout here in the stomatal crypts. That's a really good one right there. So there's two right kind of next to each other. So pretty slick adaptation. So this, even if there's a pretty decent wind rushing across this leaf, it's still not enough to really ramp up transpiration. And again, if you're desert adapted, you want to try to minimize how quickly water leaves your body. Here, they're just showing the idea of wind versus still air. The wind is in black. And so on the x-axis, you have stomatal aperture. You know, if you've done any photography, that's kind of the same idea, the aperture of the lens. And so if you have a large aperture, that means your pore is pretty wide open on the stomata. And so you can see that the transpirational flux, when they say the word flux, it just means the rate of how quickly, in this case, water is moving through the body and out through the leaves. And so if you map that out, doing still air versus moving air, you can see a huge difference in terms of transpiration rate. And so this is, a nice set of data to really drive that point home. Wind really does speed up. And then also thinking about temperature of the environment around the plant, you have data here just trying to demonstrate that with higher ambient temperature, you have the ability for that environment to hold more water in the atmosphere. All right, it can hold more water. So saturation, water vapor concentration on the y-axis, temperature, and you can see that it's a nice strong correlation, positive correlation. So as the temperature rises, the water holding capacity of that atmosphere is increasing. So it actually allows for faster transpiration rates up to a point. Same with wind, by the way. There's like, eventually there's like a critical point where the plant kind of gives up and says, that's too much wind. If you think about windstorms, for example, plants tend to not have their stomata open. With wind, they usually say wind over like 30 miles an hour, and most plants start to close their stomata no matter what. And because it's just too much, and it does start to really impact transpiration rates. Same with temperature. At some point, they're going to decide to close their stomata and conserve water, because usually they start to dehydrate at higher temps. So there is like an upper limit where the plant will hit that and have to adjust. All right, so we've been talking about all this transpiration, this movement of water through the body of the plant. And then it's like, well, it seems kind of weird, especially when they measure how much water is leaving the plant body. It's an insane amount percentage wise. it's the vast majority of the water the plant takes up goes out through the stomata. It's going right through them and out. And when they started taking all these measurements of all these different plants, they're like, wow, over 90% in some, 80% and higher in others, high percentages. It's just lost. And so it makes them like, why would a plant bother to do that? There must be some advantages for plants to have most of the water exiting out of their body through the day. So can you think of a reason why would plants transpire? What would be an advantage? Think about today. It's hot out. Think about even hotter temps, 95, 100 degrees. You're rooted in place. It's hot. What would you want to do to try to cool down? What would your body try to do? Sweat, right? You start sweating, and then those water molecules on your skin start to evaporate off your skin, and you feel that cooling effect. So that's actually a huge reason why plants transpire. It helps modulate their body temperature. Similar to you getting stuffed in a room at 100 degrees, you're going to start to sweat and try to maintain a cooler body temperature. So certainly that plays a big role. But, not all plants have to endure seasons with hot temps. So there should be some other reasons why they're coursing water through their body in such a major way. So when they take up water from their roots, what else is coming along? Nutrients. Yep. The faster you can transpire, theoretically, more nutrients are coming into you. And so it's a way to feed yourself, your minerals. There's been some discussion of, with the transpiration process, could there be maybe some toxins the plant is trying to get rid of, and it kind of gets into the water stream and out? Maybe they're looking at that. But for sure, mineral acquisition is big. And then also cooling yourself. Those are really great ways. And then, of course, as soon as you open your stomata during the day, you're taking in CO2. So, just having your stomata open immediately kicks in transpiration. But the upside of having your stomata open is also you're dragging in CO2 and allowing that the photosynthetic process to go. So they know that stomatal movement, whether the pores are open or closed, this is a huge deal for the plant. I mean, really a big deal. And so they want to better understand that so that they can help the plant grow. If you're a grower and your livelihood depends on this, this is a big deal. And so they looked at how is the plant doing that in more fine detail. And they started looking at different leaves, different species of plants, their leaves under the microscope and zooming in on the stomata. And they saw all these chloroplasts in the guard cells. So they're like, oh, wow, look at all those chloroplasts in the guard cells. So that means those guard cells are doing photosynthesis. So that means those guard cells are producing a fair bit of sugar. So maybe it is the production of sugar in the guard cells, and that will make your Sisebes, will that make your Sisebes more negative or less negative if you continue to build sugars in the guard cells? More sugars, what does that do to your Sisebes? More negative. More solutes, more negative Sisebes. And so the plant is essentially playing with osmosis. If you have a lot of sugars building up in the guard cells, that drives your Sisebes more negative, which will drive your overall water potential more negative, which means you are creating a situation where water can flow into the guard cells. And so water will flow in from surrounding areas across the membrane, and you'll get water pressure building up. that causes the guard cells to move away from each other and the pores reveal. So they went, yep, okay, we got it. And they started to, pat themselves on the back and said, that's what's happening. They were partly correct. Then a scientist came along and said, what, wait, I found some plants that don't have chloroplasts in their guard cells. They're just not there. And then they looked more at other species and they're like, oh, yeah, he's right. No chloroplasts. So now what could be going on to allow for the plant to play with Sisebes and get it more negative so that water can rush in by the laws of osmosis? So they started hunting around for that a little bit more carefully, doing some biochemical work with what's going on inside the guard cells. And what they found is that it is in the morning, if you think about plants in their natural habitat, the sun comes up, strikes the plant body, the plant gets the signal to go ahead and open stomata to start doing photosynthesis. So then they started looking for what's happening in the guard cell at sunrise. And they were looking at, is it sugar? And it's like, well, it can be to some extent, but they realized it was they were a little off track there, and they started looking more carefully and they realized it's actually potassium that kicks in first at sunrise. And so the plant essentially creates a situation where potassium loads in to the guard cell, and that messes with the Sisebas, drives it more negative, which makes the overall water potential more negative. And we know water moves from less negative to more negative. So water moves from a less negative area right near there through the guard cell membrane into the guard cell. And you get that water pressure. And so during the morning at sunrise, it's really potassium that is doing that manipulation, more and more potassium showing up in the guard cell. Then they see as the plant continues doing its thing during the day for photosynthesis, then they see sucrose is starting to play a role like at midday or, late morning into high noon and into the afternoon. Yes, then sucrose definitely is helping to drive that Sisebes more negative. So basically it's allowing for continual water flow into the guard cells to make sure that the guard cells are moved away from each other, and then the pore gets revealed. And so that's what they were tracking. Here's a visual of that, just trying to show the black line on top. So time of day on the x-axis, and on the y-axis, you're looking at the aperture. So how wide open is the pore. And so the black line, you can see that it's wide open around high noon, and then it starts to close down as the day wears on into the evening hours. But you can see this spike in potassium early on, and then that starts to fade away in the afternoon, but then sucrose takes over. And so it's just a way for the plant to very quickly, as soon as sunlight strikes their body, they have a fair bit of potassium in their body if they're have good access to that in the soil system. So it's pretty easy for them to stuff all that potassium. Potassium's small, highly mobile. It's not a big deal. They can just get that potassium dumped into their guard cells, and then sugar eventually can take over. And so keep in mind that stomata, all through the day, they can, like, this is mapping out experimental data that they did, but then you have to realize that a plant is constantly assessing its environment. So it can decide at high noon to close its stomata if it feels, so to speak, it's reading all those environmental cues. And if it's like, you know what, I'm getting kind of droughty here, I think I better conserve water. And so even in the middle of the day, they can close their stomata. It's not always like this. So just be aware that they can have really good control on their stomata through the whole day in terms of how open is that pore. Another thing that they noticed is that the level of carbon dioxide inside the plant body, especially the leaf tissue, is constantly being monitored by the plant. So remember, out in the air, the CO2 levels are pretty constant. And so that's not really a worry, so to speak, for the plant. what they're doing is assessing how much CO2 do I have in my leaves? How much do I have? Oh, it's running low. So if there's like, oh, my CO2 is running low inside my body, it makes sense that they want to open their stomata wider, or maybe they were closed and so they open them. But get those pores wide enough open so that you get more CO2 in. And so they're constantly monitoring that. And when they do lab experiments, they can show very fine control. If CO2 levels drop inside the leaves, the stomata open wide and get more in. So it's not really what's going on in the air around them. They don't really, I mean, that's pretty steady, day in, day out. Yes, it's going up because of climate change, but in general, what the plant is really concerned about is I need to know how much I have inside me because that's what I'm feeding in for photosynthesis, right? So they need to get that CO2 to the chloroplasts. So they're constantly assessing how much CO2 do I have cooking along in the photosynthetic process. So that's a big one. They're paying attention to that very much. And then light, of course, obviously, the plants around here aren't going to bother opening their stomata in the dark. And they won't take in CO2 in the dark because of that. So they won't do photosynthesis. Certainly water status plays a role on their stomata movement. If they're in a drought, they may make the decision to go ahead and close down their stomata and wait for better times. And then temperature too. Really high temps, they may close their stomata because they're starting to dehydrate. Transpiration's too fast and they're starting to get water stressed. So the movement of the guard cells and stomatal movement in general is usually how they phrase it, stomatal movement. It's a big deal. Here you're looking at the chloroplasts nicely showing up in these images. But again, initially they were all confident it was sucrose or sugar causing the movement to drive the Sisabes, but now they know it's potassium at the start. And then in terms of the sunlight striking the plant body, if you think about sunlight, and we will quite a bit in a little while here, you can break sunlight down into different wavelengths of light, red, orange, yellow, green, blue, indigo, violet. And so they wanted to know, is it all the wavelengths of light that the plant is responding to, or just certain wavelengths of light? And so they did those experiments where they just hit the plant with different wavelengths of light, and they waited to see what would happen with the guard cells and the stomata complex. What they were showing here is that you really get a spike in terms of the stomata really open wide if they're hit with blue light wavelength. Okay, so blue light is really causing a nice uptick in the aperture. So that means that a larger aperture means the stomata are more open. And so the plant is not paying attention to all the wavelengths of light to make the decision, what should I do? Should I open my stomata or not? It's the blue wavelength. And the reason it's so, narrowed into the blue is that they have a photoreceptor in their cells that is able to interact with blue light energy. And that's what they're trying to show here up in the top of the image. They're showing blue light hitting, and this is a photoreceptor called phototropin. It's right there. They have the word phototropin. We'll talk about photoreceptors in a while, several weeks out. And so it's that quality of light that the phototropin can interact with. And that I'm not going to go into details on this because it's quite a process. I just want to show you that it's complicated. It's quite the process. And this is what they call a signal cascade. And blue light kicks it off. So blue light striking the phototropin, and then you see all these arrows, right? Arrows coming off of that. And so they're trying to show that this is a signal cascade process in the guard cells. And this is not unique to guard cells. There's signal cascade processes in your body right now going on. It's everywhere. But here, for this particular case, when blue light strikes the phototropin and sets off this process, you can see potassium coming in to into the guard cells. So that's in a nutshell, that blue light allows for potassium to build up in the guard cells. And that's really what I want you to get from this image. We're not going to go into the details of that. Okay. So lots of effort and research over the decades here on guard cell movement, because we know it's super critical for the plant to be able to control that. Here's a kind of an overview again of water flow through the plant body. Unidirectional from the soil into the roots all the way up, and then finally exiting the leaf as water vapor. Okay, so transpiration is a big deal. That lots of research going into that. And if you are working with plants, it's a good process to really understand, so you know how to treat your plants the right way to optimize their growth and the yield of whatever you're trying to get them to do. Okay. Ready for the next topic? We need to get into Phloem translocation a little bit, because tomorrow in lab, you're going to do an experiment on that. So I want to introduce some of the big ticket ideas here. So we're sticking with the vascular tissue. So transpiration was all about the xylem. But remember, vascular tissue is clustered. You have xylem and phloem clustered together. So now we just kind of go next door to where the phloem is located. And we're asking what is going on in that whole highway system through the plant body. And the fancy word for that is phloem translocation. Translocation just means movement from point A to point B. And if you are a plant and you're busy doing photosynthesis in your leaves, well, you have to share that somehow. It's not just your leaves that need that sugar in order for you to survive. All of your cells need that. All your living cells need that, including parts of your body that are hidden from the sun that can't do their own photosynthesis. So potatoes are a great idea to think about when you imagine a plant needing to get sugars in order to build potato tubers, or lovely, squashes of different kinds. You need to have those sugars delivered to different parts of the body. And can you imagine if you're a grower and you depend on growing potatoes for your livelihood, how your potatoes get built? You better have a good understanding of that so that you can maximize yield and get a good profit at the end of the growing season. Anything that you're harvesting off a plant and you're trying to understand, I want to get good, healthy tissue, lots of it, so I can do what I want with it, sell it, get a good paycheck. So this has, again, been a huge area of research and trying to understand this process. Now, phloem is more delicate looking under the microscope. It doesn't have that secondary cell wall. That's why it looks so delicate. So here, this is blue-green area. This is the sieve tube elements, the phloem. We're looking at an angiosperm here. And so we're looking at sieve tube elements and then their companion cells. And they don't have secondary cell walls. They're living cells, highly modified, but living. So that means we have to deal with cytoplasm, not all the organelles. Some of the organelles are missing because the companion cells help with all that, but they still have stuff in the way. And so the plant physiologists were like, oh, okay, how do we get movement through that? That's a little bit more complicated than just the empty cells of the xylem next door. And here you're looking through a tree, 3-year-old tree, an ash tree. Here you're seeing phloem again. It's usually bluish green to my eye, but again, more delicate looking cells compared to all the rest of this image is a kind of reddish brown. That's all xylem. Here you're looking at a vascular bundle. And They call this the monkey face design. It actually can look like that under the microscope. And you can see here that you have in the so-called forehead region is where you find the phloem. But in all of these images, you have xylem and phloem close together. You have clustering going on of the vascular tissue. And I think more about Mr. Bill when I see that image, which I don't even know if you all know about Mr. Bill. That might be, you're too young for that, but that's what I always think of when I see this, especially a drawing of it. But under the microscope, it can look very similar to that. So they had a chore to do. They're trying to figure out, how does sugar water move through the phloem? And the physics of it, trying to measure the rate of it, and then trying to figure out, how is a plant deciding how much sugar to put toward like ordinary roots versus how much sugar should go into that potato tuber, how much sugar should go to that baby leaf that's growing. I mean, that's a lot of decisions, so to speak, that the plant has to make. And how in the world are they doing that? Well, a couple of things that we need to think about besides the fact that they're living cells. So here they're trying to show that with showing that there are some organelles and membranes that are in the cytoplasm that are in the way of flow. However, I do want you to notice that the artist is correct in this regard. They didn't put any membrane, at the end here. So these end plates, you can see they're creating like small, they call them sieve plates because they look like a sieve. And that's a pretty typical design especially with angiosperms, but there's no membrane in the sieve plate area. So you can see that at those pores or tunnels, if you want to think of it that way, those are all open. It's just flowing. You have no obstruction. Okay, so material just flows from one sieve tube to the next. They don't have to bother crossing a membrane at the end at the sieve plate. That's important. So keep that in mind. And so they had to determine that. They were very interested to know if that was a thing. And so here you can see how unobstructed these sieve plate pores are. So again, they had to factor that in when they were looking at movement and what forces and physical laws are driving this movement. They needed to know if there's membranes there, and they found out there were not. And that makes a big difference in how they started looking at what's going on inside the sieve tube element to allow for movement. Here you're just seeing, don't forget about the companion cells. Here's another image and cross section of the sieve tube elements. And remember, it's a 1-to-1 relationship. For every sieve tube element, there's a companion cell, right? So we have to think, they tend to think of them as like a complex, like they're kind of like a thing together. So here, characteristics of some of the elements in angiosperms. And you see number 2, sieve plate pores are open channels. That means there's no crossover membranes in the sieve plate area. And then you can see number 4, companion cells are the sources of ATP. Just getting across the point that those companion cells are really not only doing their own physiology for their own needs, but they're giving some of that to the sieve tube element, so it stays alive and can focus on just moving substances through the sieve tube elements. P protein is on here too. P protein, let me explain a little bit about P protein, number 3 there. So P protein and callus are substances made by the plant basically to help with, if their sieve tube element gets damaged, injured, they can make these substances and help kind of heal the wound. It's kind of like, when you get a scratch on your arm and your body starts, scabbing. It's kind of like that idea. They notice there's different types of pea protein depending on the species, but basically it seems to help them deal with injury. So of course you can imagine, a bunny rabbit coming along and chewing on the plant, maybe that punctures the sieve tube element, and that would not be good because of course then you're messing with the flow through there of important substances. So having a way to plug up the injury, and we actually see, you're gonna learn that that's also important because it helps maintain hydrostatic pressure that's developed inside the sieve tube element. And we'll talk about hydrostatic pressure more. Callus is another type of substance that is being used again for injury. And it can happen, it depends on the species, what strategy they do, but it seems like callus sticks around a little bit longer, like pea protein will get recycled. Callus seems to be a little bit more permanent plug to the injury and kind of stays put. But anyway, so they were looking at, kind of the fine details of what's going on with the sieve tube elements and how are they structured? What do they do if there's some injury? Then they said, okay, we're getting some basics on some of the physicality of the sieve tube elements and companion cells, but what's actually flowing through there? They suspected sugar, but they needed to experimentally verify. So it's like, well, how do we do that? Now you have to remember, this was back in the day when the technology wasn't like it is today. So this is actually brilliant for the time period this was being done. Someone said, why don't we stick an aphid on the plant? And if you know anything about those pesky aphids, they're awesome. Their in terms of their mouthpiece, being able to just very, they target so well the sieve tube elements, the phloem. They can stick their mouthpiece right into the phloem precisely. And then they start sucking away what's in the phloem. And they knew that. And so they're like, well, let's stick some aphids on the plant. And then they collected what came out the other side of the aphid. So this top image on the right there, that drop is what their waste coming out after they feed on the stuff in the phloem. It's actually called honeydew. Isn't that such a nice sound for waste from an aphid? And so they did those studies and they started analyzing the honeydew drops. Can you see a problem though with the honeydew drops as a proxy for what's in the sieve tube element? Do you think it's a really good match? So what you eat and what comes out your rear end is not a good match, right? It's kind of the same thing. So this honeydew has been altered, of course, from the original substance they took in because they used it for nutrition, and then they're excreting out the waste that they don't need. So it's very skewed data, and they quickly realized that. They're like, yeah, this isn't the best idea. So then someone said, well, we can still use aphids. Let's get them to plug into the sieve tube element and then anesthetize them and then cut their mouthpiece away from the rest of them and then just collect what's coming out of their mouthpiece. Now that's actually genius. So here you see the mouthpiece into the plant. Here they're cutting it in the middle image here, B. They're cutting it. Here you can see it's cut in figure C. And then they start collecting what's coming out. And so that's much more true to what's really in the sieve tube element because it's just traveling a little distance of their stylet mouthpiece. So those were some of the early studies. I thought that was pretty cool, creative way to approach it for the time. Anyways, here's an upshot of what you see in the phloem, and you can see sugars win by far. But there's other stuff. So other organic molecules, but also some amino acids, even some minerals like magnesium and potassium. Okay, so it's definitely not just sugar, but we tend to talk about sugar and really focus in on that. Now I'm going to jump ahead a few slides because I want to get into a little bit of the process. So when you go into lab tomorrow, I'm going to skip how they figured out it's sucrose. That's the main sugar. We'll get back to that. what they started to realize when they were looking at flow of sucrose through the phloem is that it flows from what they call sources, and then it flows towards sink tissue. So sources and sink is what we need to think about. So when they say the word source, they're thinking about most of the time plant leaves. Plant leaves are what we call net producers, net exporters. They make so much sugar by photosynthesis that they have extra to spare, so they can export very easily. And so if you think about leaves that are really active, mature leaves in particular are usually really active and are able to make a ton of extra sugar that can go elsewhere. Zinc, on the other hand, that part of the plant body is a net importer or consumer. Either it can't do enough photosynthesis or it doesn't ever do photosynthesis and needs the sugar. And so, of course, roots would be a great idea to think about, but even things like stem tissue, developing organs of all kinds, they need it to go through their respiration, to stay alive, do their physiology. They need to build cells and tissues. And sometimes the sink tissue is a way to store up sugars for, like over winter. So sources and sinks is how we describe it. And I'm going to get into kind of I'm going to go to the actual model that they came up with. All right. So this is looking at what forces are getting built up and allow for the transport of sugars from source to sink. And it's modeled after what Dr. Munch proposed. He called it the pressure flow model. Definitely pressure involved here. And what they realize is a bulk flow idea. Remember, bulk flow, the Poisson equation is delta P over delta X. You're thinking about a pressure gradient from source to sink in this case. And they did do enough experimentation that they said most of the water that they're thinking about, all the movement of water causing this pressure, most of it's coming from the xylem. Not all of it, but most of it. And again, that's kind of nice because xylem and phloem is right next to each other. So it's very handy to take some of the water and dump it into the phloem in order to generate this pressure flow situation. So let's think about the leaf. Here in this part of the drawing, we're zooming in on source tissue, an active leaf doing photosynthesis. So the far left is the xylem. Okay, you have the xylem right next to the phloem here. Now you have the companion cell in blue mostly. They're showing the companion cell is kind of the bluish cell. And then the sieve tube element is right next door. Okay. And then this source cell here, this one, that is like a spongy parenchyma cell. Okay. So the spongy parenchyma cell is the source cell. It's doing a ton of photosynthesis. All those yellow balls are the sucrose that eventually gets made. And they're showing the flow of sucrose to the companion cell and then to the sieve tube element. So you are effectively messing with psi sub S. That's what you're doing, right? You're dumping a bunch of solutes, sugar, into the sieve tube element. That's going to make your psi sub S more negative, which means your overall water potential starts to go more negative compared to cells around you or the xylem next door to you. And so then you'll start to have positive water flow. Water flows from less negative. Here you can see water over on the left in the xylem. You see a water potential of negative 0.8. And you see a water potential in the sieve tube element, it's more negative at negative 1.1. Water moves from less negative in the xylem to more negative in the phloem. So that dumping of solutes drives your psi sub S more negative, overall water potential more negative, water rushes in to the sieve tube element. Now you get a positive pressure building up. Okay, you get positive P, which they're showing there with psi sub P is equal to 0.6. Okay, but that's half the story. You have to finish the story. So, okay, we get some nice pressure up in the sieve tube element in the leaf. Great, but what's happening at the sink? You have to think about this happening simultaneously. You have loading of sugar in the sieve tube element up at the source. Let's imagine this is a root tissue of a potato plant. where the sink cell is a developing tuber. All right, let's think of it that way. So this sink cell is the potato tuber that's growing. We need to get sugar to that so it can keep maturing and growing. So you have to unload the sucrose. And we actually say that word. We're unloading the sucrose out of the sieve tube element down in the sink tissue and getting it to the baby tuber. So, okay, well, if you have sugar leaving, exiting out of the sieve tube element there, what's happening to your Sisabes as sugar gets out of the sieve tube element? What's happening to the Sisabes in the sieve tube element? Less negative or more negative? Less negative. It's becoming less negative because it's losing all those sugars out of it. So it's less negative now. Okay, so by the laws of osmosis, water will also start exiting out of the sieve tube element to go to a more negative area. And so here, what they're showing is a lot of it goes right back to the xylem, because if you look at the water potential of the xylem next door, it's, whoops, 0.6, negative 0.6. Whereas in the sieve tube element, it's negative 0.4. So less negative to more negative. So you see the water is exiting out of the sieve tube element. Well, if you have water going out, that's going to lower your pressure. So your Sisap is going to drop. And that's good because now in the sink area, you have lower pressure. In the source area, you have higher pressure. You got a gradient. So now you've created a situation where you can have sugar flow from source to sink based on a pressure gradient. So it's a very simple mechanism in play that is creating a gradient of pressure, and that's allowing for the sugar movement. So putting it all together, here's the image in full, source on top, sink on the bottom, and they're showing the sugar movement from the source to the sink based on a pressure gradient. Now, there's one weird thing I want to point out. Let's just focus on the sieve tube element. You see water potential in the source is negative 1.1. Water potential at the sink is negative 0.4. But we said water's moving down to the sink. But if you just look at water potential, you'll say, but wait a minute. Sink is less negative water potential. Source is more. Water should move up based on the laws of water potential. But I just got done telling you, no, it's a pressure gradient and it moves down in this image. So what you need to remember is that when you think about water potential, we're talking through a selectively permeable membrane. And I made a big deal about there's no membrane that water's crossing in that sieve plate area or anywhere else. So once you have water in the sieve tube element, there's all open channels. And it's just a pressure gradient that's causing this movement from source to sink. Make sense? So when you think about the laws of water potential, there's a selectively permeable membrane involved. And you start thinking about that. But because once you're in the sieve tube element, there is no selectively permeable membrane. And so you get just a pressure gradient driving the process. So in lab tomorrow, you're actually doing a fairly simple setup to demonstrate this pressure gradient idea. And the equipment you'll use, Duncan will talk to you about how, different parts of it represent different parts of the plant. So you're going to be doing that tomorrow, and then you'll also be setting up two of your hormone experiments. So it's going to be a little bit of a couple things going on tomorrow. Okay. All right, that's it for today. We'll continue on with Phloem translocation on Friday. So next Wednesday will be the